Welcome back to part 3 of chapter 2 on the chemical foundation of life. In part 2, we have just gotten done with talking about all the different unique properties of water. And we ended with talking about pH changes, acidic solution versus basic solution, and how buffer can resist the changes in the pH. So the last section of this chapter introduces the concept of biomacromolecules, and what you will see as a common thing among these molecules is that they are usually made up of a lot of carbons, carbon as in the chemical element. Previously, we call the atoms that come together as molecules. Well, when many different atoms come together to form a complex molecule, we call it a macromolecule, such as a DNA, RNA, proteins, carbohydrates, and lipids that we will introduce and talk about in detail in the next chapter 3. And all of these macromolecules makes up a big component of our cells. Now, the fundamental component of macromolecule is the element carbon. And remember, we learned in the beginning of the chapter that carbon is capable of forming 4 covalent bonds to 4 other atoms. And this usually ends up serving as the structural backbone of many of our macromolecules. The simplest molecule formed by carbon and hydrogen is methane, which is made up of 1 carbon and 4 hydrogens, and they form a 3-dimensional conformation called tetrahedron. So this is carbon right here, and these are the 4 carbon, and this particular geometric shape is called a tetrahedron. Many larger organic molecules can be made up of entirely of just carbons and hydrogens, and we call those molecules hydrocarbons. And hydrocarbons are what we oftentimes use for fuel purposes, such as propane in our gas grills. And because all of these, why we tend to use hydrocarbons end up being able to use them as fuel is because all those covalent bonds between each hydrogen to the carbon can contain a lot of energy. And when that bond or covalent bond is broken, it can release those energy that is stored within. And that's a concept we'll talk about in chapter 6 for metabolism. Okay, so these bonds right here, other than representing the electron that is shared between the 2 atoms, if we were to break these bonds, it will release energy. Now, hydrocarbons can exist in different 3-dimensional geometric shapes, and one that we have just introduced for methane can be in the form of tetrahedron, or they can be linear carbon chains or carbon rings, or both of them can exist. And carbon-carbon bond can be a single, double, or even triple covalent bond. We're talking about C single bond C, C double bond C, or C triple bond C, okay? In these type of molecules. If we are talking about chemicals with 2 C's and then the amount of hydrogen it has. So this bond between 2 carbons can be in that single bond, double bond, or triple bonds. And all of them contribute to the molecule's overall 3-dimensional shape that is critical to their function. As I've said with proteins, structure is to function. If you change the structure of something, usually it means you're changing the function of that molecule as well. So those hydrocarbons that tend to be linear in structure are called hydrocarbon chains. So that can be easily remembered. Chain are usually linear. Like a metal chain. So that's why they're linear molecules, and they usually are connected by successive bonds between the carbon atom. So we're talking about, lots of C's connected to each other, and then, hydrogens attached to every one of those carbons. so on and so forth. I'm not going to draw the whole thing, but sometimes those carbon atoms can be branch or unbranch. So this, what I've drawn here, is an unbranched version of the hydrocarbon. But if we're talking about something like this, then this is a branch version of a hydrocarbon chain. Okay, branch means it's completely linearized. Branch meaning maybe, a branch coming off like a T shape. Okay, so those carbons that tend to be linear in structure, like I said, are called hydrocarbon chains. And single bonds within hydrocarbon chain allows for some flexibility, such as this or this, between the molecule itself, while double bonds like this will make the molecule more stiff in terms of structure. So single bonds allow for a lot of rotation, which allows for some flexibility in the molecule, while double bonds don't. Okay, so this can be applied to the concept of unsaturated versus saturated fat that we talk about when we get to lipids in biomacromolecules in chapter 3. Hydrocarbons that exist excuse me, in a ring structure are called hydrocarbon rings. And again, they can be made up of single or double bonds as well. So here is an example where the hydrocarbon ring is just made of single bonds in these two examples. And these two examples contain carbon-carbon double bonds within the hydrocarbon ring itself. And some examples of hydrocarbon rings are, for example, benzenes that are a lot of times incorporated into amino acids, cholesterol, estrogen, and testosterone. Again, we'll talk about these macromolecules in the next chapter. Another example where the macromolecule can contain both hydrocarbon chain and hydrocarbon ring is beta-carotene here, where you can see at these two ends is a hydrocarbon ring that is connected to each other by a region of hydrocarbon chain. So hydrocarbon chain, hydrocarbon ring, they can exist by themselves or as combination within a macromolecule. And while we are on the topic of chemical geometric shapes, some chemical molecules can share the same chemical formula but differ in the placement of atoms or chemical bonds. And these are called or known as isomers. There are three main types of isomers that we will talk about in this particular chapter. And you'll learn more different isomers that there can exist in chemical molecules in chemistry, I'm sure. So structural isomers are chemicals that has the same chemical formula. Okay, so same amount of C's, same amount of H's, but they differ in the placement of covalent bonds. So an example of this is where both of these molecules shown here has the same chemical formula, C4H10. They both have 4 carbons and 10 hydrogens, but when it is either in the hydrocarbon chain format, unbranched, versus hydrocarbon chain branch format, they're considered as 2 different chemical molecules. One is called butane, one is called isobutane. And even though they have the same amount of C's and H's, they have very different chemical properties because of how differently they are shaped. Butane is fuel that we use in lighters, fire lighters. Okay, so the liquid within there is a fuel that is made up of butane molecules. Isobutane here, which is this branch version of the hydrocarbon, is actually a refrigerant. Okay, one is for fire, heat, while the other is a coolant, okay, as a refrigerant. So they aren't very different in chemical formulas made up of the same C's and H's. All they differ is in their structure. So that's why I say structure is very important in the function of chemical molecules a lot of times. Okay, so that's what structural isomers refer to. Same formula, but different placement of the covalent bonds. Okay, structural isomers. And then you have a second type of isomers called geometric isomers, which are similar in the placement of covalent bonds, but differ in how these bonds are made to the surrounding atoms. So, for example, we have both C double bond C in the middle. Both are C4H8, okay, having 4 carbons and 8 hydrogen, but both are called butene molecule, but one is called cis-butene molecule, one is called trans-butene molecules. Whether they are in the cis or the trans configuration has to do with where the methyl groups are. Okay, methyl groups are referred to as CH3s. So in this cis-butane molecule right here, both of the methyl groups are on the same side of the carbon. There's a bulky CH3 group. Because what I mean by bulky is, technically speaking, if we expand this, we're talking about 3 H's sticking out like that versus just one little H here. Okay. Whereas in the trans configuration, you have a methyl group here, okay, and a methyl group on the opposite side of the double bond. One is on the same side of the double bond, one is on the opposite side of the double bond. Being on the same side is called cis configuration. Being on the opposite side of the double bond is called the trans configuration. The cis configuration is usually involved making a bend in the molecule. Because these two here are bulky groups. Okay, so if we're talking about C single bond, and then all of a sudden there's that double bond C, and then C onwards, it tends to sort of bend it this way. Okay, instead of a linear molecule like this, it creates some kind of a bend in the molecule itself. Whereas trans, when it looks like this, there's that double bond C, but it exists as more of a linear like molecule when it's trans molecule. Okay, which gives us the idea or the concept of unsaturated fat versus saturated fat. Unsaturated fat, we're talking about a form of triglyceride that is in cis conformation, where there will be a bend in the backbone, carbon backbone. And trans configuration, in trans fat, like this. So this is an example of trans fat. This is an example of cis configuration, or what we call unsaturated fat. Okay, so, and that's why trans fat is bad for our health, is because in our bloodstream, trans fat being more linear for conformation like this, or structurally like this, is more likely to pack tightly with each other to form solid chunks of fat inside our bloodstream, such as formation of plaques, which increases our chance of cardiovascular disease if that plaque was to form, or blood clot starts forming and dislodge into your heart or into your brain. Okay, so if it goes to your heart and start blocking blood vessels within your heart, it's going to cause a heart attack. If it's a clot that goes to your brain, then it will result in a stroke. So that's why trans fat is, quote unquote, unhealthy for you, because they are linear molecules. They can stack on top of each other like this and form a whole block of solid fat within the blood vessels, for example. Whereas cis or unsaturated fat, they have that little slight bend in their structure, so they're not, less likely to form stacks on top of each other because of that bend, and they usually remain liquid or oil at room temperature. Which is why if you were to ever see some trans fat products, they tend to be solidified at room temperature, while unsaturated fat tend to remain in that oil liquidy conformation at room temperature. All right, and the last type of isomers in this chapter that we'll mention is the enantiomers. Okay, so we have structural isomers, which are different ways that the atoms can be bonded to each other. Geometric isomers, which we're talking about cis versus trans, where the arrangement of the different groups or of atoms can differ between two molecules. And the last thing here is enantiomers, where again the molecules have the same exact chemical formula. The amount of different atoms is the same. The placement of atoms is about the same, but the molecules are actually superimposable mirror images of each other. Meaning that, for example, if you have your left hand versus your right hand, and if you try to superimpose them on top of each other, you put this hand over this hand. It's kind of hard for me to show you without you seeing me in person doing the superimposing, but try putting your right hand on top of your left hand, okay? And you will see that, yes, you have your thumb, okay, and your pinky on both hands, but your thumb ends up being on top of your left hand's pinky, and your pinky of your right hand ends up being on top of the thumb of your left hand. Your thumb doesn't go on top of thumb. Your pinky doesn't go on top of your pinky, okay? So that's what we mean by mirror images, because if you look at your hands like this, okay, they are mirror images of each other. Thumb here, and then pinky here, pinky here. But if you move this hand to be on top of this hand, the thumb then becomes on top of the pinky becomes on top of the thumb. They are not the same they're not the same structure, okay? Just like here in the case where you have a carbon attached to a fluorine, a hydrogen, chloride, and bromide, okay? Fluoride, bromine, and chloride. And then you have another carbon here that is attached to a fluorine, bromine, chlorine, and hydrogen, okay? These two molecules are mirror images of each other. But if you were to move this C on top of this C, the C's would align with each other, the F's would align with each other, but the bromine would end up with the hydrogen, and the hydrogen would end up being on top of the bromine. So that's why these two molecules are not exactly the same as each other, but non-superimposable mirror images of each other that we call enantiomers of each other. All right, and that's now we're down to our last section or last little slide for this chapter, and this is a glimpse into the next chapter of biomacromolecules of life. We introduced the concept of functional groups here. Functional groups meaning the chemical functional groups like these. We know what we talk about as methyl groups we talked about earlier, or hydroxyl group that we talked about as earlier. Remember I mentioned hydroxide ions. So wherever we see OH, we refer to that as hydroxyl groups. So These functional groups make up our biological molecules that will allow the molecules to have different specific chemical properties for their unique function. Some play a role in structural cases, and some play a role in chemical reaction cases. These play an important role, for example, in forming DNA molecules, your proteins, carbohydrate, and your lipids. For example, your carboxyl group, and right here, COOH, or your amino group, NH2, okay? OH, NH2. This is referred to as the amino group. This is referred to as carboxyl group, but they are main functional groups that are important in the component of amino acids that makes up proteins, as we will see in the next chapter, is what defines our amino acid structure. One side being having a carboxyl end, one side having an amino end, and then they will participate and interact with each other to form a chain of amino acids. And that's all I have for you for chapter 2 on chemical foundation of life. We won't dive deep into this functional group until the next chapter in application of the macromolecules, and we'll continue that in chapter 3. Okay? All right. See you.